lecture is now being recorded, and this is what we saw last time. We plotted the raw signal, so y-axis has this arbitrary units of signal and x-axis is time here. The orange one is the control signal and the green one is the actual signal, which is which we are interested in. So we saw that we use one of the signal as control signal, which accounts for artifacts like movement or any other kind of noise that may be present and common to both the wavelengths that we shine on the fluorophore. So only one of the wavelengths responds to respond to dopamine, but the other wavelength is independent of dopamine. So it will cover contain only the artifacts or the features that we are not interested in, basically. So you may be able to see here around like this time that there are some kind of uh, patterns that are common in isospastic signal as well as excitation signal, the slow uh, patterns or even faster patterns. If you see by zooming into it here, so there are some kind of jumps which are present in isospastic, which are uh, also present in excitation. Not always. So if this peak is there, this peak is only present in excitation. So this is probably the peak of actually dopamine release inside the brain. So but we want to control for so what we are also interested in is the percentage jump in this fluorescence at the time of dopamine release, and that will come only when we control for the baseline. So the baseline control can be done by isospastic wavelength. So these are the raw signals. I will also apply some kind of filtering. So that filtering is nothing, but for now I am only applying median filter and a low pass filter, where I am removing the high filter noise, by high frequency noise, basically. So we expect our signal to behave in certain range, and that's why we can apply a so I am applying a filter of like 30 hertz, because I don't expect my signal, my uh, like dopamine levels to be faster than that, to fluctuate faster than that. So I am plotting that, and next what we did was, okay, I will next time I should on present from my lab PC. This my personal laptop is actually not good, so I will close the other figure. We got this these are denoised signal, which are like high frequency noise has been removed here, and now you see they are like thinner, but this is the part that that Deepak covered in last classes, the how these band pass filters work and what all goes into it. So we are more interested in the next steps of this analysis. So in next steps, what we did was we applied a high pass filter. Basically, it allows only higher frequencies to go in. So that was to bring all this signal on a zero baseline level. Both of them, both of these signal. So to it can also correct for photo bleaching, and then we applied linear regression to correct for movement artifacts. These were our next steps. So, but for now, what I will do is, instead of applying high pass filter, I will just visualize the raw data first. So in visualizing raw data means I am trying to figure out that is there any way where in which we can see a a correlation between the excitation signal and isoasbestic signal. Basically, the signal that is interest to us and the control signal. So if you see here, this particular plot, it says that there is a clear positive relation between isoasbestic FFI, which is denoised, and FFE, denoised excitation. So it means that most of our signal, no, most nahi, but at least a part of it is correlated with isoasbestic, and we can predict uh, s- some part of excitation signal uh, using our isoasbestic signal. So what does this mean? That the motion artifacts come, which also show up in isoasbestic, can be determined. We can determine them using isoasbestic and subtract them from excitation to get true signals. But can we do that subtraction directly? So I will show you that by some example, like what are we doing next. I will come to what does each point here represent. Each point is a just one data point collected at the same time point. So let's say you collected so let's say you collected excitation data at 0 millisecond, and then isoasbestic will collect around uh, uh, 2-3 milliseconds after that, depending on our like signal, our hardware. So that we consider a single time point data, and then next reading will be somewhat 7 milliseconds after that. So we collect this excitation and isoasbestic in pairs basically. So at one time point we collect excitation, then isoasbestic, then excitation, then isoasbestic. So these pairs are very close in time. So we each point is that uh, readings at that time point. Would it be right to say that uncorrelated points are our signal of interest? Yes. The uncorrelated points are our signal of interest. Yes, that is true. So you see this even in this plot. Um, there are two things to note. One is the scale of isoasbestic is around 158 to 172, and the scale of excitation is from 160 to 180. So any change in isoasbestic can't be directly subtracted from excitation because the scales are different. So we need to find that line not only by subtracting but through some other method, right? Means which can take into account by this scaling, and that method is linear regression. We will see how that method comes up. And another point to note is that this if you see, there is a line here, but there are a lot of positive values above that line probably, right? This is just from raw data, denoised data visualization, that we can tell this. So this is a known thing, that this excited the dopamine rises positively, more than it, like the the peaks in dopamine are higher than the valleys that come up. So this is a known fact. Like this particular sensor also can't detect the dips. It detects the higher concentrations better than the dips. So this is a function of both dopamine signaling as well as our sensors, that you see that a lot of positive signal is present instead of a negative signal. So Vinod, what was your question?
What are the units here? Are we sort of measuring the concentration of dopamine?
Correct. So these units are arbitrary units, and basically you are measuring the luminescence of reflection of the emission that you get from the brain when you shine some light. So that luminescence you can measure using a camera, using a, photodiode, or you can use PM photomultiplier tube, anything. So a sensor will give you some reading. Depending on the sensor, you will get that unit. But for now, you can call it an arbitrary units as well. But this may happen that uh, the level of isoasbestic readings that you get out from the uh, sensor is not same as excitation. Basically the scales may be different, so that may be because you are shining lower intensity isoasbestic light, you can't always match that intensity of shining also, right? So there are always some gaps. So that is not a problem, but that is a thing that we need to take care of for in our analysis. So what we want to do next is um, uh, how do we find that line or that relation that uh, what part of excitation signal can be explained by isoasbestic signal. So that is something which we get from linear regression. So I'll open my presentation if it works. Okay. Okay, so we are at this fit and subtract and divide state. This B and C we will see today. So I will show you one simple example for this. Let's say our true signal is of this shape. Okay, this is my true signal. Okay. Uh, but for whatever reason, when I measure signal, I get something like this. Okay, so this is something is unknown. This is not known basically, and this is measured signal. So what we are doing here is we wa- we don't know the true signal. We want to figure out the true signal using the measured signal. So we have a control signal here. That is the isoasbestic, and we know that isoasbestic will behave something like this. Should behave something like this. So this is thing. Now let's say if both of them are centered at 0, then what happens that you can just directly subtract ms minus cs and you will get your true signal, right? But in general what happens that both of them are not centered at 0. If you are not applying high pass filter before regression, then they are not centered at 0. And what you need to do is you need to apply some scaling to this control signal. Basically you will apply this control signal is isoasbestic, FFI, and measured signal is excitation, what we call is FFE. So now what you do is instead of just directly subtracting, if you, let's say you are measuring it at level of 100, and this fluctuation will be, so you, this fluctuation may be of range, let's say 25 in your excitation signal, but in your control signal you are measuring at level of 10, and this can be like 2.5. So here you need to multiply them by 10, and then subtract. So you will get true signal by ms minus 10 into cs, right? Or there can be some kind of offset in uh, offset itself, like where you uh, multiply something and then that add that offset. So basically that means that if your excitation signal is fe, control signal is fi, then you estimate it as a linear line m into fi plus an intercept, where m and c are obtained using simple linear regression. And then motion corrected signal, we get it as fe minus fe estimate. So what is this linear regression that we'll see today? So what you are trying to we do here is an ordinary uh, is what we are trying to do is estimate this m and c. So what values of m and c should I choose? Can I choose them arbitrary, or is there a criteria for them to choose? So one criteria is, I will draw on my iPad. I'll stop recording from here. Okay. This would not. Regression, which is, so this is x and this is here, this is our y, this is our x. And so what we is there somebody in the Himanshu, do you have a question?
Yeah, hi Atharva. So my question is, what is the reason behind this offset difference? Uh, it,
So that
does
thing
it have to
can
do with
come
the?
up because, see, the sensor itself may have different emission properties for different wavelengths. If you shine exact same intensity of wavelength A versus wavelength B, the emission you get will be different, even in absence of dopamine. Second thing is the intensity of lights that you shine, they are also different. So you actually, this is a calibration step that is done before every recording, that we calibrate the intensity in microwatts, basically what power of light you are shining for both the wavelengths, and we try to match them. We try to match them such that our signals are around the same, our output signals are around the same intensity uh, levels. So these are like two reasons. I hope you got it. Okay, so what we are doing is let's say we measured something here, 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 right. So we are trying to find this particular line which is not known to us. So even let's say we get what we do in simple linear regression is that oh, is that we take these error terms. This is E and this is also E. Oh, this is beta pen. So for any line that you draw, these are the error terms that you will get for any arbitrary line that you have drawn through that space. These are the error terms that you get. So one way to draw this line, that okay, this is my particular line, is get these error terms to 0. Like, or minimize them, not 0, but minimize them. But what happens here is that the error can be positive or negative, either positive or either negative. So there can be many lines which can achieve you this particular thing. So you can even draw a line like this, which will give you all negative errors here and all positive errors here, and that line will give you error of 0. So this error can be this is not the criteria that we use in ordinary least squares. Why is it called ordinary least squares? Is because what, instead of minimizing the sigma E, you are minimizing sigma. So you take this error from the line and you minimize that. So if these are your points, you draw any arbitrary line and you minimize these errors. So what this line is called is a y cap line, basically. So here y cap is equals to beta naught plus beta 1 x, and our y will be beta naught plus beta 1 x plus some error term y I e I. Okay, this error term is in for individual sample points. So what we are minimizing here is this sigma ei square, and that minimization gives us a line that fits the y and x value, and that line is called a line obtained through ordinary least square regression. So it's ordinary least square estimate. So this is what we do, and this beta naught and beta 1 value, so this is just a scaling factor, right? It's just I was telling you that a scaling factor and an offset that you use. So this offset is very simple. Basically you take the average point, y average, and you match them at average value, basically x average. So beta naught will be nothing but y average minus beta 1 cap x average. So this is my beta naught, but for this you need, of course you need to know this. So if you minimize this quantity, you will get beta 1 cap as covariance of x comma y divided by variance of x. So this is what you get for beta 1 estimate, and the beta naught estimate is this. So this is basic theory, some formulas that come up. But uh, ma- main thing here to understand is how do we interpret this. So one kind of interpretation is that if you plot this error terms for a good fit, if you plot this error terms with respect to y, okay, so for a good fit, or actually let's say if what we are trying to do with the linear regression is that we assume that the population is uh, uh, linear related linearly related in y and x. Basically the population itself has some kind of uh, formula that y is equals to beta naught plus beta 1 x. So now you see I am not using a cap on beta naught and beta 1 because these are true population measures that we cannot ever estimate, and that is an epsilon. So what we do here is for for ordinary least square estimate we don't assume, but uh, for in general what we assume is that this y are, so this is let's say x and this is y, and this is our beta naught plus beta 1 x line. So y's are epsilon 1, epsilon 2, epsilon 3, like that. Okay, so these points denote y, and what we assume that this epsilon is normally distributed about 0 and some sigma. But this assumption is not required for ordinary least square estimate. If this is true for population, if this assumption is true, then our ordinary least square estimate becomes the best estimate for our y that we can get. So there are other terms like maximum likelihood estimator or UMVUE, those kinds of terms are there. So I will just call that as best estimator. When this assumption is true, our OLSE is the best estimator. On a population level. And if even if this is not true, even like in any case, OLSE converges to the true beta naught and beta 1 when n tends to infinity. So when the number of sample points tend to infinity, this OLSE estimate, the estimate of beta naught cap and beta 1 cap, that we saw from the previous formula, these estimates, covariance of x comma y divided by variance of x, beta naught is y average minus beta 1 x average. So these estimates converge to beta naught and beta 1 when n tends to infinity. So these are the properties of OLSE estimators basically. Now with this info what we can do, like these are all theory, okay, so what we can do is we can visualize at least. So what we do is we plot error terms with respect to y. So these error terms for a good fit should look like evenly distributed from a positive and negative direction. They should be like this. Okay. And this spread of E means they can be as far as away, because let's say if you are you don't have a good estimator for your y. Let's say y can be your ice cream sales and x can be temperature of the day. So when you estimate using temperature of the day, you can get residuals, but uh, they may be wide. This width may vary. Let's say you estimate using humidity instead of temperature, then again this estimate may vary. But if it is normally distributed, then it is close to noise that you can get. Basically, it is noise of the system itself. You can't do so. This is basically this epsilon. Epsilon is normal about 0 comma sigma square. So if sigma is high, if the in population itself, which is a which we are not estimating, which is a property of the system, that the numbers will vary about this line with some variance. So that inherent variance is high, then your error bar will be also far away from zero line, and they can be less away from your zero line. So this spread only doesn't tell us if our fitness, our fit is good or bad. We need to scale this with something. So what we use as our uh, like measure for uh, like how useful is our model, basically how useful is this beta naught cap plus beta 1 cap x model is, we use a term called r square. So this spread in itself, what can be denoted by sigma of ei square, right? You have already minimized that for a particular line. You have minimized this. So what we are doing here is, if you say that okay, for one particular variable sigma ei square is like one particular fit, like a last session's fit, let's say my last year's data gave a sigma ei square of 100, but my this year's data is giving me 200 minimum, like minimum of sigma ei square was 100 in one of the samples and 200 in another sample. Can you say this 100 when the sigma ei square is low, is this fit better than this 200 fit? Actually we can't, because they are scaled on a different level. So what you need to do is you need to divide it by the variance of y itself. So what we do is, the r square formula basically is 1 minus SST by SST. Okay. So what is this? These are very tricky terms. They are used in as a different like nomenclatures are there in different books. But what is this SST and what is this SSE? Sorry, I have written it opposite. So we want to estimate that what is this how good is our model fit. So what we do is we take this SSE, basically it is sum of ei square, and we take SST, that is sum of y minus y bar square. Okay, and we divide SSE by SST. So for a better fit, SSE should be low, right? The sum of square should be low. So we subtract it by 1 from 1, and this is also equal to SSR by SST. So what is SSR? SSR is sum of अरे oh So this is sum of squares due to regression basically. And uh, SST is, so you can call it as variance explained by uh, your x, variance in y explained by x. This is total variance in y, and this is unexplained variance. It can arise from any sources. There are other x's probably which you have not considered. For our data, that x may be dopamine level. So you are only considering isosbestic signal, and you are trying to uh, explain the variance in the signal that you are getting. But the major contributor might be the dopamine levels, and that you are not taking into account. So that everything else will go into unexplained variance. So what our r square give is, r square is this 1 minus SST. So this can give you a good idea that how good your model is to be used. So this is how E plot should look like. You can take sum of E square, but that sum of E square should be scaled with variance of y to uh, to basically be to interpret that, right? So not necessarily this particular fit was better than this particular fit. It may so happen that on this given day the y was less noisy than this day. So this is something that we should keep in mind, and there are other interpretations for this kind of residual plots that you should have in mind. So if you plot
Expression for SST.
what
Expression for SST.
y minus y bar square sum, so it's the variance in y. So total variance in y, out of which you can explain some part of it using your regression, that is in blue, and out of and whatever is rest is unexplained variance. So you can actually do the math and find out that this add up to summation of y minus y bar square in our OLSE. So whatever you get here and here, you can add this up and you get this y minus y bar square. Okay. So let's say if your spread of error terms go something like, instead of like this, it goes something like this, like it expands as y increases. So what mean about the data? How was the data looking like? So one plot I will draw first, and then I will ask you to give inputs next time. So if it's my line, okay, these are my points spread like evenly across that line. But what in this red case, what may happen that it makes a funnel shape basically, like this. So these kinds of fit are like they change in x and y. So when x increases, the variation in y also increases. So here in our ordinary least square assumption, what we assume that this sigma, basically the spread of population, is constant. But it may so happen that this sigma is a function of x. And if it is a function of x, let's say it is increasing, then you will get a bigger bigger errors here and smaller errors here. So it will not be a straight line fit, but so in this case you can't solve it by whatever I have told you right now. So what you have to do is you have to keep this sigma square as a variable. The function of sigma square also has to be estimated, and then only you can estimate beta naught and beta 1. So that is something complicated and we'll not go into it today, but this is why the plots of E versus Y are important whenever you are doing uh, ordinary list square regression, any regression basically. So this E can also behave like this, and you may now probably tell that what should the plot look like. Anybody wants to?
Opposite funnel, like
Yes. Yes.
contracting.
So, like opposite. Oh, sorry. You got it right. Huh. This eraser is bad. I should buy an Apsara one next time. Like this. There are other forms of error terms that you can get. Oh. Okay. Other forms may include an error spread of like this. Does anyone has any guesses for like what is the problem in doing a linear regression when your error terms look like this? What it tells you is that at certain values of y you have almost zero errors, but as you go above and below it, your error increases. So can like do you have some idea like how does my y versus x look like in this case, and what are the remedies that I should follow for these kinds of situations? So
Maybe X and Y are not linear.
yes. So this indicates that there is a non-linear association between y and x generally. So let's say if your plot y versus x is distributed like this, and your OLSE will predict a line something like this. Okay, so your errors at these points will be minimum, but as you go up higher it may increase. So you may also see like something like these patterns. For other kind of polynomials you can see that kind of pattern. So this is something that you should look for, right? So what we want from our error terms is to look like this. When we plot with y, they should not increase or decrease, they should just remain at 0. So when you get this as a perfect white noise, when you get this E spread around 0 as a perfect normally distributed function, let's say normally distributed variable, then there is nothing to be added on. Like you will have covered every possible variable that is there. It's only pure noise that is left. It is pure white noise. Then there exists no better model than what you have done, you have made. So uh, that is the goal. But since you don't consider all the models in your all the variables in your model, often you will get a vary some kind of variance in your E versus Y plots. These kinds of variance may be like more of positive errors than negative errors. Those kinds of things may arise uh, because in our let's say if you perform that on the example I took, if you regress this particular signal with this, so then you will get a more positive errors than the negative errors, and you might be able to subtract this, and like you might be able to regress and subtract, and then you get a prediction of this red curve as this, and you subtract both of them. Basically you put a minus sign here, and then you get the true signal. So this uh, uh, this may give you a more positive errors than negative errors. That's the point I wanted to convey. So I will show you in the data, and since we started late because of some technical glitches and my PC, I will not be able to cover curve fitting, the exponential curve fitting. That is a very not a very large topic. I'll try that, and I will show you these residual plots for my own data basically. So I'll come back to my PC. So what does linear regression mean? So using linear regression you can also fit exponential curves. The difference is that the equation should be linear in your coefficients, not in x. So even if you write y equals to beta naught plus beta 1 into e to power minus x, let's say e to power minus x something in x, then also it is linear, right? You are still estimating beta naught and beta 1, but when you are, let's say okay, so you can do similar kind of uh, concepts from regression in curve fitting also. You can use that. So what happens here is that, let's say you have a equation like this. So this is a double exponential fit. This comes in handy when we want to see how our signals are bleaching, like we want to control for that. So our this fluorescent signals have this property of photo bleaching, where if you keep shining light, the output will decrease over time because of some chemical changes in the fluorescence property, physical and chemical changes. So this kind of photo bleaching is generally modeled using a double exponential fit. A into exponential minus at plus b into exponential minus bt. So let's say if you have been given this small a and small b by some by your sensor company, and you only need to estimate a and b from your data, capital A and capital B, then also it is linear regression. Now we have two variables, at and e to power minus bt, which are known function of your variable time here, and a and b are still linear, right? So this is again, this is an estimate, right? So now here our independent variable is time and dependent variable is y, So y estimate is a exponential minus at plus b exponential minus b. We have to estimate capital A and capital B. But there can be other kind of problem where the small a and small b is also not known. So you have to estimate all four of them. That is not a linear regression problem. Now that has become a non-linear regression problem. So this non-linear regression problem do not come up, generally do not come with mathematical fixed solutions like we did in OLSE. Like, okay, beta one value is covariance of x comma y upon variance of x. So that kind of solution don't exist for non-linear regression. Generally, you can make them. There are many mathematicians and statisticians working on that, but generally you don't. So you solve them using uh, numerical methods. Basically, you try them, hit and trial methods. So you try different variables and find and converge to some solution where these uh, again the sum of square of residuals basically is minimized. So your goal is to minimize sigma e square for a given y. Yeah, the goal is to minimize sigma e square divided by variance in y. But since y is constant here, your goal is to minimize sigma e square. So let's say I will show you some example data here. Uh, this is focus on column A and column D for now. Okay. So this is my signal uh, with time. This is how it looks like, and okay. So on y-axis is the signal and on x-axis is the time. So this is kind of double exponential signal. I have to fit it a double exponential plot to it, where I have these four variables: capital A, capital B, small a, small b. Small a, small b are the time periods of time constants of basically tau 1 and tau 2 of different exponentials. So let's say I will start from some 0.7, 0.3, 0.2. These are my initial estimates. Okay. So these four values, I made a guess that okay, they are 0.7, 0.3, not 0.3 but 3. Oh, actually 0.3. And small a and small b are also 0.3 and 0.03. So these are my initial estimates. So I put the fit as the formula for fit as this: capital A into exponential of A here minus A into time plus capital B into exponential of small b into time. So this is my fit value, right? Fitted value. So now I will apply this formula to all the numbers down here, and I will get some fit value. So for next step, I will calculate ei square, which is nothing but y minus the fitted value. This is my fi, the fitted value, and this is yi. So basically fi is y cap I. You can also see that here. So this is my error terms squared. Okay. So I have applied that formula already, and this error term squared I will call it as SSE, sum of error term squares, right? 8.36. So my goal is to minimize this, but to make it interpretable I will also write an R square here. So I will first of all get y average. So y average is nothing but average of the signal, overall signal. So I have taken that average. Then SSE I have already calculated as sum of ei square. So SST, again SST is nothing but sum of column G, and column G is what? Yi minus y average square, basically the variance. So SST is the variance in y. R square is 1 minus error square divided by um, variance square. So this is the percentage of variance explained by uh, my fit in the signal. So I have to maximize this. So what I do, I will show you using a plot. I will include select data. I'll add a series. So I'm adding time on the x-axis, and the name is fit, and on y values I am adding the my fitted values. Okay, so I am trying to plot my fitted values on the same plot. Fitted values are in column E. Okay, so this is my fit, right? This orange one is my fit. You can see that. So what? But this is not optimal. This does not is not very a very nice fit to look at. So what some numerical methods can do is they can try different values of these four variables and minimize our SSE. This can be minimized or this can be maximized, like whatever you do is same only. uh, and these have to be varied, right? These four variable have to be varied. So now any numerical solver can do it for you. The Excel also has function, and in Python, in my notebook I have used a scipy curve fit function. So there are different numerical methods that you can use to solve these kinds of problems. So there are whole uh, uh, like courses on numerical methods. So I have already added a solver here. Basically what I am trying to do minimize is this value here. Then I have added the variables as these four variables. Okay, I will just so r square is for current, my initial guess is 53%, and I have done some, like applied some constraints also on my um, so this solver input I have applied some constraint that this should be less than or equal to 1 or and greater than or equal to 75. Like I needed a model that can explain this kind of things, and I can actually not have that constraint at all. I will just get less than or equal to 1, and I and then I will just run this solver. So this is just to show you that how what kinds of steps are involved. You can just run this in one single line in Python. So there is some model working on it, and it gives us some signal as it does not converge. So basically that constraint, if I apply, so for numerical methods you have to give a right uh, initial point to the, so right initial guess to the solver, and also choose the right methods to solve it. Otherwise they may throw out errors, or they may give you some local optimum also. So you have to be very careful in that way. So
Do we know that our initial values are not right?
so for that, if you see, go to my code, I will go to my code, that what kind of initial value I have used. I will show you that. So that is a general recommendation in the field, like what kind of value initial value you can use. So those initial values depend on y. So if you have a double exponential fit, you can express this in a function of y base initialization, that you can take, okay, average of something something or 50%. So I will show you that. So I'll just try to solve this again. This may not work. The the algorithm that Excel use may not work. Okay. So now it is uh, improved my fit. So you can visualize this. This is more looking more like uh, fit which is closer to what we had guessed initially. So you have to apply the right constraints also. That is important in these kinds of solver. And our R square has increased from 53% to 74%. And this is uh, how you check. So I can also plot here EIs basically equals to signal minus fit, and I can plot a histogram of it. So this histogram like looks perfectly kind of normal, and this is because the signal I have chosen is like that. Basically the signal is nothing but these are the true values for the signal. So I have taken 0.6 into e to power minus 0.2 t plus 0.4 into e to power minus 0.02 t, and these are my true values, and I have added normal noise to that. That's why you get that EIs as perfectly normal, and these are our estimates. So these are how, like, this is how close you can get. They are still not 0.6 and 0.4 because of our sampling that we have done. It's only 450 points. As you take, so these are only 450 points right now. If you take this to infinite points, then these numbers will go to 0.6 and 0.4, even if my noise is not normal. That's what this oh, that is in linear regression, but this is non-linear regression. So this is already a not an OLSE thing, but for OLSE this will go. So for non-linear regression there are many solvers that are available, and this is one example that I wanted to give you, like just to manually go through it, that how curve fitting is done. So I'll
Sonor
huh?
was it?
I will show you here now what kind.
Which
Yeah, yeah,
Sonor?
yeah. Tell. So this is how our what I give as, sorry, initial parameters. So you can study this function basically, uh, curve fit. So this curve fit is a function in scipy.optimize, and it takes some the signal basically, uh, and it takes this double exponential is a function that you define, which take time, then which takes a constant, then which takes a amplitude of fast component, basically capital A, then capital B, then small a, small b. Uh, Not even small b is obtained by tau multiplier. So these are just quirks of individual function. You can have a tau fast as small b also. So that's okay. Uh, but you give initial parameters. So these are the initial parameters that you give. So max signal by 2, max signal by 4, max signal by 4, comma, uh, like this is the time window. 3600 is the maximum time that I run for my sessions for. So you should see what is this initial parameters function for format for this particular curve fit function, and that way you can give this initial parameter. So uh, what I have chosen is maximum, half of the maximum signal, then 1/4 and 1/4 of maximum signal is my initial parameters. So this particular curve fit has this recommendations, and then you can get an exponential fit to your plots. and I'll show you this is the last thing probably for today, and then we will disperse. So yeah, here I will show you one more thing. I will jump between the linear regression thing and this. My laptop would have worked. It runs in like seconds, or taking too long a time now. Okay, so it's not able to plot. My laptop is not able to handle this call with the code I figured out today. I will probably start the next class from plotting this. Oh, we plotted something at least, because it is not defined. I'll just try one last time. So in this step I am applying a high pass filter. It plotted a figure. Okay, good. And then I will do linear regression. Okay, so this is the I am doing linear regression here, and it actually looked nice. So this is what we discussed right before starting today, that so these kinds of the slow changes that are visible both in control signal and excitation signal should be subtracted basically. So that we do with our linear regression thing. I will close this. Okay, it is plotted now. Okay, so this is how uh, like y, this is y on our y-axis excitation signal, and x-axis as the isoasbestic signal. So this is the predicted FFE and denoised FFE basically. So we will subtract this prediction, and we'll also see comparison between what is the prediction and how the actual signal looked like. I will just show you here. Not responding. Okay, so we'll go to around same time probably here. Yeah, so you remember this kind of patterns that we were seeing in isoasbestic. So now this is not isoasbestic that I am plotting. I am plotting the prediction from isoasbestic, basically linear combination. So it will have that particular shape. That is obvious. That it is just a linear function of that isoasbestic. So if we subtract this value from the green curve, we will get the true signal. That is the whole point from here. And you can now visualize the residual plots, which I don't have time for now. And I will also show you where this exponential fit. So in the end, what signal you get is a delta f, basically how far is the your f from 0, that you can get from by applying a low pass filter and then high pass filter, and then everything will be centered around 0. But that increase in signal is also assumed to be proportional to the actual value of signal. So if your f was high, then that signal increase will be low. So, oh, sorry, high. And if f was low, then that signal increase will also be low. So in the end, you do a delta f over f, and for that baseline f, uh, you need to have that exponential fit. So basically, that's what I am doing right now, and that's what we that's why we saw that exponential fitting thing. How does that work? So if it plots, then it's fine. Otherwise, so this is how exponential fit looks like. So now this green curve's exponential fit in black, you see here. So this will become our baseline, and now everything like delta f over f, when we say, then this is the f that we choose for that delta f. So this is how we use this exponential fitting here, and I also showed you like how does the background, like how does the algorithm work in backend of it. So yeah, so this mostly covers the photometry part. We are not left with much. I can probably start with electrophysiology in next classes. So this is it. Stop sharing, and if you have any more doubts about it or something, if you want notebooks, I can share that those with you personally, if you want to play with them. Otherwise, we are done with photometry, I guess. That's it. I will just confirm like if anything is left, and I will let you know by mail over this weekend. Next two classes will also be taken by me, I guess, and I will tell you what I will cover. Most probably we'll start with electrophysiology. Okay. So thank you for attending and asking questions. I'll end the class. Bye bye.